I'm looking forward to tell you something about immune repertoire sequencing and our own company, ENPICOM. So ENPICOM is a software company located in the Netherlands. And in our company, we gather a diverse set of expertises that ranges from, for instance, immunologists to bioinformaticians and software engineers. And what all these different figures and profiles have in common is an in-depth understanding and knowledge about repertoire sequencing. So first of all, what is repertoire sequencing and why repertoire sequencing might be something interesting for you? So repertoire sequencing is a technology that allows to use next-generation sequencing to profile the adaptive immune system. How does it work? So on very high level, you start with a biological specimen that could be blood, it could be culture cells, it could be tissue. You prepare the samples, there is a specific preparations, we target specific amplicons in these samples. Then when the sample has been prepared, then is moved to sequencing with a normal standard Illumina sequencing machine. Then a lot of data then is generated usually through these experiments, and that data is then analyzed with a software solution that we realized to analyze immune repertoire sequencing and give what we call an immune profiling of your original samples. But then what is this immune profiling that we're talking about? So this immune profiling is a characterization at the nucleotide level and a quantification of the B and the T cell receptor. Specifically, what we provide is a detailed overview of what is the sequence of the hypervariable region. So the region that encodes the CD3, which determines the antigen specificity of these receptors. On top of that, we provide sequences, but also abundance. Therefore, we are able to give an overview of what are the expanded clones in your samples. So which kind of analysis can you perform and which kind of insights you can have if you have this kind of data available? First of all, since we provide sequence level information, you can compare receptor sequences. And you can compare these receptor sequences in your internal study as well with external studies. If you compare the receptor sequences internally, you can, for instance, imagine that you can track clones in longitudinal studies. That means that if you have a vaccination, that you can have samples before vaccination at different time points through vaccination, for instance, at day 7, 14, 28. And what you can do, since you know the sequences up front, you can follow them through time and see whether they expand and which one expand and how much these sequences expand in your sample, as well as when they recontract. So you can see the expansion profile of your vaccination study, for instance. Similarly, you can also look at historical data and see whether you encounter the same clones, for instance, in previous vaccination studies or across different patients. That's the power of having the sequence available. Furthermore, if you look at the actual external studies, you can see, for instance, if specific clones, which are elicited by a therapy or by a specific vaccination, are present in different diseases, for instance, or have been found by different studies in different labs. And that also allows you, for instance, to identify whether your clone might be also interesting in a different disease, as well as you can eliminate some of these clones from your analysis if you already know that belong to infectious diseases in which you are not interested. For instance, clones which are specifically related to CMVs or EBV or other infection that might be undergone to that same patient that you're treating at the same time. If this information is available in, for instance, public databases that contain annotations on all these clones or several clones on their specific, for instance, epitopes that they target. Now, this is a kind of analysis that is possible to do with repertoire sequencing, but this is sort of still a basic analysis that relies on comparing sequences with each other. However, in the context of two clinical trials that we are running, one in France and one in the United States about cancer vaccines, we are applying more sophisticated analysis. And in this respect, there are two specific analyses that we are applying. One is called TCR specificity clustering. What this analysis does, of course, you can imagine repertoire sequencing give you hundreds of thousands of different clones or different receptors in your data. Therefore, there is a question of which clones recognize which antigen, then of course, or which kind of PMHC peptide complex. Now, the question is, of course, we cannot answer from the sequence of the TCR what would be the specific antigen, but what's possible to be answered is which set of TCR present in the data have the same specificity. So which group of different TCR, for instance, we recognize or is most likely to recognize the same PMHC ligand. This is, of course, very useful when you're tracking clones through different longitudinal studies, because you're not anymore tracking a single clone, which might undergo several fluctuations in your longitudinal studies, but you're tracking a cluster of clones which have the same specificity, so therefore equally interesting for your treatment purpose. So this is one analysis that we perform, and we combine it with a second analysis, which is called generation probability. This relies on the concept that not all sequences, or let's say not all recombination, have the same probability to be generated. So using different sets of V and J genes and different P or N nucleotides in the junction give a different probability to generate a specific recombination, so a specific receptor sequence. And some of these receptor sequences are more likely to be found in a random repertoire, and others are less. And the underlying idea is that sequences that have low generation probabilities, which means that are very unlikely to be found randomly, if they expand through time, that means that probably there is an antigen pressure to make such that such clonal population expands. And that's what we are trying to combine together with clustering. So the kind of analysis that we perform, for instance, to monitor vaccination studies, is that we perform repertoire sequencing analysis before vaccination, after vaccination. We compare these two sets of samples, and we try to identify if there is a clonal population that expands and expands because it's under antigen pressure. And these plots, I will guide you through it to just let you understand which kind of insights you can get. So you have, of course, that you have a lot, each dot in this case represents a TCR cluster, as we defined it before, so cluster that have the same antigen specificity. As you can see, most of the cluster lies on the 0 point here, which means that most of the clusters are not expanded in between before and after vaccination. The size of the cluster represents in how many different patients that cluster has been found. So what's called the publicity of such clones. And the positioning on the x-axis shows how if a cluster expands in that direction, for instance, after vaccination, or contracts in this direction after vaccination. While the y-axis here represents the generation probability, actually the minus log of the generation probability. What that means is that clones which are more likely to be found in repertoires are on the bottom of this plot, and clones which are unlikely to be found in a random repertoire and found on the top of this plot. So what you're looking for in such an analysis are clones which are located towards the top part of the plot because they're unlikely to be found, and they are expanded, something like, for instance, this cluster over here, which look rather expanded and as well rather unlikely to be found randomly. Now, an interesting aspect that you can already observe by looking at this plot is that most of the large circles, and I remind you, large means found in multiple patients, are towards the bottom part of the plot, which also is congruent with the idea that those are sequences which are more likely to be found randomly. Because if a sequence is more likely, a specifically recombined sequence is more likely to be found randomly, therefore is more likely to be found in multiple patients, so to be public. And that's kind of what you can see here, seeing that most of the clusters that are larger, therefore found in multiple patients, are also most of the more probable cluster to be found. Therefore, potentially a cluster like this means that it's found in multiple patients, yet is quite unlikely, yet is expanded upon vaccination. Something interesting to follow up, for instance. Now, this kind of analysis, of course, require a certain amount of expertise, both in terms of managing the data, as well as to perform this kind of bioinformatic analysis. That's why, as a company, we developed the IGX Platform, which is an end-to-end platform which takes as an input the sequencing data, the raw sequences file, and is able to manage this and analyze and visualize this kind of analysis that I showed you. So you don't require bioinformatic expertise. It's a sort of bench task application that biologists can use without any specific expertise in the computational part. One of the core value of the platform that we developed is the ability to link metadata and experimental data at specific clone level, which means that you are able to associate, for instance, spot or early spot analysis to specific clones and have all the kind of different, for instance, avidity or affinity analysis that you're performing if you have that available at the sequence level, as well if you have annotation regarding to the specific epitopes recognized by the single clone. And that allows you to do powerful searches through external databases. Because if you have this annotation that a specific clone appears in an external database and is annotated with an epitope which is related to CMV, you can search through your internal study and identify whether there are clones that match that external information. Then another aspect of the platform is, of course, the ability that is modular. So there is a core part of the platform that allows to actually handle the data and to manage the data, but there are several modules that allow to perform specific analysis. We have a module for quality control, which is very important. It's called IGX Inspect, as well as a module which does the annotation, because you need to go from the sequencing reads to the clone annotation, so extract, for instance, the CDR3, the CDR1, and all the different regions on these clones, which is called IGX Profile, as well as downstream analysis, like, for instance, CDR3 length distributions, and other analysis of this kind, which is called IGX Explore. And of course, we are developing more modules, and specifically a module, for instance, for tracking vaccination studies and monitor vaccines. And therefore, in this case, if there is anybody that is interested to explore these possibilities and to be heard and to understand what are the problems in this situation, we will be very happy to welcome you at our booth, which is just next to the door, to discuss further about this. And I will be happy to take any question.